Okay, for topic 7.1, you need to be able to talk about that DNA. Your entire genome, right, is a whole lot of DNA. You've got 2 meters of it in your nucleus, and that's broken up into 23 pairs of chromosomes. And within those chromosomes, there are different types of sequences of nucleotides that make up that DNA. So we've got places that are actually highly repetitive, where you have a nucleotide sequence repeating itself over and over again, like TACCG, TACCG, TACCG. Those highly repetitive sequences generally don't have much of a function that we're currently aware of, but they are quite useful if we're doing DNA profiling. And we'll be looking at that when we're doing um, our biotechnology unit. There are also protein coding genes, which we're most aware of. We'll be doing with transcription and translation. And then there's other parts of DNA that we consider structural DNA that has some important roles, but it's not actually technically in a protein. So the highly repetitive sequences, it's between 5 and 45%, depending on the person, the genome and the chromosome. And it's usually a pretty big chunk of base pairs, 5 to 300 base pairs. And we don't know the purpose, but they are transposable elements. They can copy themselves within. But in the end, the biggest use we have for these are in DNA profiling, because different people have different numbers of repeats. So like you might have 5 repeats for this particular section of DNA, and someone else might have 2 repeats in that particular section of DNA. And we can use those number of repeats to be able to determine who did it at a crime scene. It's a little more complicated than that, but we'll get into it a bit later. All right, protein coding genes, we know these. These are the single copy genes. They've got coding functions. So like there's a gene for making hemoglobin B. There is a gene for making rhodopsin. So these are the genes that we know of that translate into, we will transcribe and translate into proteins. This is actually generally less than 2% of our chromosomes. And within the fragment, within the genes themselves, there's actually these exons, which are the coding parts, and then there's non-coding parts called introns. So if you take an entire length of a gene, some of those parts actually are used to code for the gene later, get turned into mRNA, and other parts are actually introns that ended up getting spliced out at some point. But they do have an important function, because sometimes one gene can actually be translated differently because of the exons and introns. You'll see that when we're doing transcription. So don't stress about it right now, but just think about that when we're doing the transcription part, that these introns are actually important non-coding parts of DNA that can be used by our bodies or by our cells when we're making proteins. Okay. So telomeres. What are telomeres? Telomeres are pieces of DNA at the end of the chromosome. So your chromosome is a certain length, and at the very tips, both tips of the chromosome, is a section of DNA called telomeres. These are important because when you're doing DNA replication, you can't actually replicate like the last few nucleotide sequences. So that means every time your cells replicate and divide, your DNA is actually getting shorter and shorter and shorter. And they actually think that that's part of the aging process, why some of our, as we start to get older, some of our cells are starting to deteriorate, because our DNA is shrinking. The telomeres add some buffer to that. So the telomeres, as it gets shorter and shorter, your telomeres are getting shorter, but it's not cutting into those DNA sequences. And so a lot of the research right now is for anti-aging is looking at how can we actually get our telomeres to remain the appropriate length, so we could maybe overcome getting older. There are other non-coding segments of DNA that actually help in gene expression. They can regulate it, so they might help promote gene expression from happening, or they can sometimes actually like stop it from happening. So they can regulate it by either promoting it or promoting and enhancing it, or it can actually stop DNA replication in other ways. And then we have, of course, genes to make tRNA. And if you remember from when you're in 9th grade talking about protein synthesis, tRNA is a very important molecule that it's involved in the translation of mRNA into a protein. And so the we actually have to make tRNA, and tRNA is a segment of it's essentially RNA, right? But it's going to be made on the copy for that gets made on your genes, on your chromosomes. So those are also non-coding functions. And then, of course, the introns that we'll get into in more detail when we do um, transcription.